All right, class, so before I get into showing you how cells are using DNA, like what we're really using DNA for, I want to tell you a couple things about RNA. Right, so RNA stands for ribonucleic acid. Right, so very similar to DNA. DNA was deoxyribonucleic acid. Now we're looking at ribonucleic acid. Right, so the differences here, DNA versus RNA. I'm not going to do the like this complex structural part with RNA, but the differences are the sugar here is ribose instead of deoxyribose, and then the bases are going to be G, C, A, and U. Right, so this has uracil. RNA will never have thymine. Right, so DNA we saw T, not U, never U. And here with RNA we see U, never T. And we still get complementary bases. In RNA we still get G with C and A with U. Right, those are still complementary bases that form. Right, and there's a few different kinds of RNA, but I'm going to just show you real quick one specific type of RNA. Hey, it's called mRNA, and this stands for messenger. Hey, this is messenger RNA. Okay, so this is going to carry a message from the DNA. I'm about to show you something that's kind of an intro into how we're using DNA. Right, so this messenger RNA is part of that. mRNA will be part of what I show you. And just think of that mRNA as having a message from the DNA. Okay, now really, really important here. Our first idea of what is this message. This message is always going to be how to build a protein. That's what the message is. Right, so the DNA has that message in it already. One of the things we do is make a copy of that message, a copy of those instructions, and that copy is mRNA instead of DNA. All right, so that's what mRNA will be doing. Right, so this intro into what DNA is used for. What do we do with DNA? It's called the central dogma of biology. A central dogma of biology. Hey, this is a core theme. All cells do this. So this is the, in a nutshell, we're going to do a little more detail with this in the next video, but in a nutshell, we are looking at how are we using DNA. Okay, so it looks like this. We start with DNA. From that DNA, we make our copy. Our copy is going to be mRNA. And then we use the copy of the instructions that are in the mRNA to build our protein. So the whole central dogma, that meaning all cells, all organisms do this. You know, might there be little minute differences? Yeah, of course there are. But all cells do this. DNA to mRNA to protein. Every cell all the time will do this. Right? So, two steps here. DNA to mRNA is called transcription. Right? So transcription, it's like we're transcribing, right? We've got instructions in our DNA and we want to make a copy of that. And we make that copy as mRNA instead of DNA. And then mRNA to protein, that's called translation. right? So the central dogma of biology is DNA gets transcribed into mRNA, and then our mRNA is translated. The message in our mRNA is translated into a protein. All right, so I'm going to show you real quick then how this is going to be happening. Just a real quick example. So if this is our DNA, then the first thing we want to do with this is transcription. So use this DNA to make mRNA. Hey, and actually let me show you real quick. Let me just label real quick. Like this particular piece of our DNA would be a single gene. You know, genes are technically hundreds or thousands of bases long, so like this a gene's not going to be 7 base pairs like this. But just for the sake of doing this in a short, concise way, just think this section of DNA we're working with is a gene. So we transcribe this into mRNA using complementary base pairs. There's a G, we put in C. There's a C, we put in G. Now careful with your T's and A's and U's, right? The blue mRNA should never have a T, but it does have A's, right? So T, we get an A, we get a U, then a G, then an A, then a U, right? So those are the tricky ones, right? Because you got to make sure you pay attention to your A's and U's and T's. A lot of people, when they see T in the DNA, they'll just go, okay, no T, so put in a U. But, you know, of course, that would make it wrong. So, you know, just take a second and think about A's and T's and U's when you're going through doing this. Okay, and then we would translate this into our protein. So I'm going to show you a little more with this in a second, but what happens is each of these sets of 3, these bases in our mRNA are read 3 at a time. So these are basically, they're kind of like the words in our instructions. And remember, proteins are made of amino acids. So it would just tell us, okay, what's the first amino acid? And then the next word, UGA, that would tell us what's the next amino acid, number 2. And then, you know, again, I only made this 7, but we would have the next word to tell us what's amino acid 3. We would have the next word to tell us what's amino acid 4. So you get the idea. Right, so that's what the message has. The message has what's the first amino acid, what's the second, third, fourth, fifth, etc. All right, so that's what we're doing when we're using DNA. DNA has the instructions on how to build the polypeptide or how to build the protein, and we go from DNA to mRNA, and then we use that to build the actual protein. Okay, that's the central dogma.